We'd like to welcome Annerieke Sierksma today. We've already had kind of a wonderfully interesting lead-up discussion that I'm hoping continues through this presentation and beyond. Um, the Most of the talks previously in this series have focused on diversity in developmental models and in cerebral organoids or systems along those lines. But as we'll hear today, individual differences are quite relevant throughout development, function, maturity, and in this case, investigations of neurodegeneration. So we're really interested to see how your perspectives in a very different cell context kind of mesh with the thinking we've been doing. Thanks so much for agreeing to give the talk, and we're excited to hear what you have to share with us today, Annerieke.
Sure. Yeah, thanks for reaching out and giving me the opportunity to present. So indeed, I'll be taking a bit of a different approach. So I'll give a bit of a lean up on Alzheimer's disease and the type of research that we do and the questions that we ask. But indeed, we do look at kind of genetic heterogeneity and how that can shape, yeah, predominantly disease trajectories, and in this case, microglial function. So I do already want to acknowledge some people up front. So Sarah Bory, Dan Mugars, and Mark Viers, who've been really pivotal in helping with this project. So I want to shout out to them beforehand. Let me see how
Wonderful.
this works. Okay, so right, just a bit of a primer. I guess you all know this, but Alzheimer's disease is actually the main cause of dementia. So roughly 60% of dementia cases are caused by Alzheimer's disease. Some of those scary numbers are that in our lifetime, roughly 1 in 5 men is diagnosed with dementia and 1 in 3 women. And so that means that roughly every hour we have 5 new dementia diagnoses. And so what is actually really difficult about Alzheimer's disease and particularly in diagnosing that, but most prominently in actually treating Alzheimer's disease, is that the disease really starts decades before the symptoms appear. So actually, Alzheimer's disease first starts with the accumulation of amyloid beta pathology, as you can see in the upper right corner. And then this is followed by the appearance of tau pathology that also slowly spreads throughout the brains and starts to basically impact different brain regions. And then that is quite closely followed by the onset of neurodegeneration. And so we see this quite profound tissue loss and neuronal loss, particularly in regions involved in learning and memory, but also in executive function, in speech. And then, of course, that right when there's severe enough neurodegeneration, this is followed by all these clinical symptoms. So people suffer from memory issues and speech impediments. So what is good to know is that there's a roughly 10 to 20-year delay between the first initial accumulation of amyloid beta pathology and actually the onset of the first clinical symptoms. And so this makes it very difficult to actually treat individuals because by the time they are showing symptoms, the disease is already 10 to 20 years ahead. And of course, this really makes it difficult because then if we start treating patients all the way roughly at the end of the disease, right, there's in that sense little we can do. We can rescue the neurons that are still there. But of course, ideally, we want to diagnose people way up front, all the way in the beginning of the trajectory, and then intervene right at those very early stages, so we can kind of prevent people progressing all the way to this dementia clinical phase. So over the years, we've been recognizing there's likely different phases that the brain goes through. So the first phase is, we have coined this biochemical phase. So that's when the first amyloid beta plaques start appearing. So there's an overproduction of amyloid beta and different longer species of amyloid beta, which accumulate into these amyloid beta plaques. And then there's this so-called cellular phase, and that probably dictates how long a person has between the onset of the amyloid phase and the onset of this clinical phase. So when the person starts having symptoms. And the cellular phase is probably dictated by how the rest of the brain cells basically deal with this amyloid stress. So how, for example, your microglia deal with clearing the plaques or maybe may even help to seed the plaques, but also how good your neurons are at coping with this amyloid toxicity and at what stage they start developing tau pathology and neurodegeneration. So understanding that cellular phase will be very important, and also that probably gives us the room to basically manipulate so that we can maybe push people further away from this from the onset of that clinical phase. So over the years, there's been a lot of research on the genetic landscape of Alzheimer's disease. Of course, that gives a way into further understand it. Right on this graph, you can see very causative and high penetrant AD risk loci, which are very rare in the population. There are also some protective variants. Those are also very rare in the population. And then there's this whole slew of SNPs and other loci that have been implicated to either increase or decrease your odds for developing Alzheimer's disease, roughly only by 1 or 3%, and they are very common in the population. And so by and large, we know that those causative and very strong risk increasing variants, right, they cause familial forms of Alzheimer's disease, and they likely affect the processing of amyloid beta. And then we have this whole tail of small risk loci that probably dictate the sporadic forms of Alzheimer's disease. And they likely affect this cellular response to amyloid beta pathology. And particularly they pinpoint towards microglia, so the immune cells of the brain, as probably being very modulatory in this response. So there are a lot of SNPs that fall in genes that are either exclusively expressed in microglia or highly expressed in microglia, or those risk loci fall exactly in microglial enhancer regions. So that means that they can only steer microglial gene expression and not per se the expression of that gene in other cell types. So microglia, of course, have this, well, there's still a lot of unknowns about microglia and Alzheimer's disease, and there are different, different parts that they can play. And I think there's still a lot to learn about them. So on the one hand, microglia are probably good for the brain because they help to clear the amyloid beta plaques, either through phagocyting, either soluble species of amyloid or insoluble species of amyloid. But by phagocyting them and actually trying to degrade those in their lysosomes, they also may actually help to seed plaques because probably they can't fully degrade these amyloids, and therefore they may actually also seed new plaques. But these microglia may also form this kind of protective barrier around the rest of the brain. As you can see on the right, you can see where microglia kind of form this protective barrier, and where they form this protective barrier, you see a lower occurrence of dystrophic neurites around the amyloid beta plaques. So they may indeed shield the neurons actually from becoming dystrophic around those plaque regions. Research in our lab has also shown that microglia likely have this kind of dual mechanism in Alzheimer's disease. So they may actually actively contribute to seeding plaques. There was Nora Baligac in our lab did this experiment where she depleted microglia from an APP NLGF mouse, so a mouse model of amyloidosis. And she saw that if she depleted microglia at an early phase when there was no plaques present yet, then she actually saw that she could reduce the amount of amyloid beta plaques that there were present in the brain. So that shows that if microglia are present, they are probably helping to seed plaques in the brain. And then when she depleted microglia at a phase when plaques were already initiated, then she could see that once microglia were removed, actually the size of the plaque increased. So that likely shows that in later stages, microglia may actually help to compact the plaque and therefore provide some kind of protection to the brain. And so that, again, that directly actually demonstrates this difficulty in when we try to find also a drug for treating Alzheimer's disease. If we try to modulate microglia, we also need to think carefully about which stage these microglia are in and how we should best manipulate them to help the patient. So we also know, right, that genetics for Alzheimer's disease can really impact how microglia function. So for example, with the TREM2 R47H mutation, we know that this can actually have a negative impact on amyloid beta load. So if you carry this mutation, you probably have larger plaques, as these microglia are less able to recognize the plaque and to phagocyte them. But alternatively, actually, this mutation may be beneficial for tau-induced neurodegeneration. So also depending on the mutation that you carry and depending on the phenotype you're looking at, microglia may be good or bad. We've also seen that, so not only does the genetic impact on the microglial function, but we've actually also seen that depending on the pathology that the microglia see, that they can up or down regulate specific Alzheimer risk genes. So we could find in mouse models for Alzheimer's disease that microglia particularly upregulated all kinds of Alzheimer risk genes when seeing amyloid pathology, but not when seeing tau pathology. And we saw very similar things also in iPSC-derived microglia that were xenografted into a mouse brain. So to show that indeed all these different Alzheimer risk genes were only becoming upregulated or downregulated when specific pathologies were met. So that shows that right, Alzheimer's risk may become very apparent only when it is presented in a certain particular context. So when we want to study Alzheimer's disease, very often, and we've also done this in the lab, right, we focus on one gene and we try to understand the impact of that one gene on, right, on a particular function. So for example, on the function of microglia. But of course, we also know that our whole genetic makeup, of course, in the end dictates our risk for developing Alzheimer's disease, as Alzheimer's disease is highly heritable. And so also that this genetic risk for Alzheimer's disease spans beyond GWAS uncovered risk loci, right? And can also include things like other genetic variations, such as structural variations or repeat sequences, or deletions or other types of mutations. And so in this research that we wanted to do here, we actually wanted to kind of go beyond a single gene, a single effect type of approach, but we wanted to look at this more holistically and understand more, right? How does your whole genome basically impact on your, right, on cellular function? So how does your whole genome basically affect your risk for Alzheimer's disease? How does this translate into different cellular responses in the brain? And we had a focus then on microglia. And we also, of course, were wondering a bit on how can we research this, because it's quite a daunting question. And of course, how many people actually do this is often by looking at postmortem tissue, right? So they isolate microglia, they relay the transcriptome of the microglia and right compare that to the genome of those individuals. But an issue with microglia transcriptome is that microglia really quickly change their transcriptomic repertoire depending on the environment that they're in. So depending on if people have more or less amyloid pathology or more or less vascular pathology, the transcriptome of the microglia may change a lot as a function of that. And of course, the microglia transcriptome may also change as a function of the genetic background. So we wanted to actually devise a system where we could kind of keep that brain milieu stable so that that would be the same for all the different donors that we would investigate. And we could really understand and kind of tease apart the impact of genetics on the microglial transcriptome in isolation from the environment that they were in. And so we utilized iPSCs because, of course, the iPSCs inherit the genetic background of the donors that they come from. And so that was a great platform for us to kind of understand that genetic diversity and how that could impact the microglial transcriptome. So what we did is that we basically made use of a couple of different cohorts that were available to us. So we first used the HIPSCI initiative, which is an initiative from Sanger, right, which is this vast array of stem cells. There was also a collection of iPSCs that were generated by Tracy Young-Pierce at Harvard in collaboration with Rush University Medical Center. There's these ROS and MAP cohorts of individuals right, that are aged, and they follow these people longitudinally. And so some transition to Alzheimer's and some don't. And then also through collaborations at our local hospital in Leuven, we had access to 2 different cohorts that were also followed up longitudinally, as well as a case control cohort. And so we reached out to our collaborator, Valentina Escott-Price, who basically also researches the genetic components of Alzheimer's disease. And she calculated so-called polygenic risk scores. And so what that does is that for each individual, you basically tally whether they're heterozygous or homozygous for specific SNPs that are associated with Alzheimer risk, and you weigh them against the betas that are derived from the GWAS. And so in the end, you get one compound score that kind of indicates whether a person has a high risk or a low risk for Alzheimer's disease. And so, right, for every individual within those cohorts, we generated these polygenic risk scores. But then you can make a distribution of all those individuals. We standardized those distributions against the Caucasian population of the 1,000 genomes. And then we selected individuals really to kind of span the entire range of this polygenic risk score. So spanning from -3 standard deviations to +4 standard deviations, meaning that we have individuals that really have very little genetic risk for Alzheimer's disease to individuals that have quite high genetic risk for Alzheimer's disease. And then we selected mainly cases on the positive end and controls on the negative end. But we also took some individuals that kind of spanned that middle. So we really have a distribution of a risk for Alzheimer's disease. So after we selected all those individuals, we either acquired the iPSCs or we generated them ourselves from PBMCs. And so in the end, we made this cohort of 31 lines where we selected 5 individuals from the HIPSCI cohort. So for those, we don't know their Alzheimer's status. And then we have 13 coming from the University Hospitals in Leuven and 14 coming from the ROS and MAP initiatives. And so covering both non-demented controls as well as AD cases. So in the lab of Hortus Soper, we had Renzo Mancuso at a certain point as a postdoc in the lab, and he initiated this xenotransplantation model to study human microglia in an in vivo context. And so with the help of Nicola Fattorelli and Anna Martinez Moriana, he set up what they have termed the migrate protocol. So that's basically a protocol that can differentiate iPSCs or embryonic stem cells to microglia progenitors using a series of cytokines. So that pushes them through hematopoiesis and in the end generates these microglia progenitor cells. So what we do at day 18, we can collect the microglia. They kind of bud off from these embryoid bodies. And we harvest them and we can xenotransplant them into a mouse brain. And to be able to do that, we actually use a CSF1R inhibitor so that depletes all mouse microglia from the mouse pups. So we give those pups 2 injections at P2 and at P3. And then so at P4, the brain is relatively devoid of mouse microglia. And that creates a kind of niche so that the human microglia can go in and occupy and colonize the brain, basically. So using this model, we can, of course, inject these human microglia in different types of mouse models. So, for example, in the APP-NLGF mouse that develops amyloid pathology over time, or in a wild-type mice, that's the APP-human mice, which has a humanized A-beta sequence, but does not produce any amyloid plaques. And then, of course, we can sacrifice those mice at set times and investigate then how these microglia behave using immunohistochemistry, flow cytometry, or single-cell sequencing. And as you can appreciate, at roughly 6 months of age, you can see that every green dot is a human microglia, that the mouse brain is really nicely colonized by these human microglia. And you can also see, right, that they have this really nice ramified morphology. So here you see actually an image where right, the parts where it's black, those regions will still be occupied by mouse microglia. And you see all the human microglia in red. But you can also appreciate that they can adopt different activation profiles when they encounter this amyloid beta plaque in blue. So then you can see that they start expressing markers of activation, such as human CD9, which is a marker for kind of this disease-associated microglia profile, or also MHC class 2, this HLA-DR DQ-DP antibody that also represents a kind of activation state. This was a really nice model, and that was a tool, of course, that we were using. But we now had this daunting 31 lines that we wanted to graft, and we had actually already started grafting those individually into different mice and then harvesting those different mice for doing single-cell RNA sequencing. But then, of course, there was this great paper that came out from the Kevin Eggen and the Stephen McCarroll group, right? And I understand that Michael Wells also took over that research and took it along with him to start his own lab. Where right this idea of these cellular villages were coined, and so we thought if this can be done in vitro, why shouldn't we be able to do that in vivo? So yeah, we stepped into this and decided to try to do that in vivo, and that worked actually quite nicely. So we set up what we call the human microglia village xenotransplantation model. So where basically we have now our 31 lines of genetically heterogeneous iPSCs, and we generated 3 villages. So we had in each village, we had 12 to 13 lines. There were 3 lines that we had in common across the 3 villages. And so what we do is that actually we differentiate every line in single. So every line goes through its own migrate protocol to generate these microglia precursor cells. Then we pool them at equal ratios, and then we graft them into the brain. So we graft each pool either in wild type mice, so in the APP human mice, or in APP NLGF mice. So these APP NLGF mice, they develop amyloid plaques. And then at 6 months of age, we sacrificed all these mice, and then we isolated out the human microglia using flow cytometry, and then we sent those off for sequencing. And what we did is that we used these site-seq hashes basically to label the microglia coming from particular mice. And then we used SNP-based demultiplexing to understand, of course, which microglia comes from which donor. So that in the end, we had 235,000 human microglia. And for each microglia, we knew from which mouse it was derived. So whether that mouse developed amyloid pathology or not. And we also knew from which donor it was derived. And so for us, this was, of course, a great way to really separate out how genetics influences microglia versus how the environment influences microglia. Because what was really nice is that we could, of course, directly, compare each donor side by side, because each donor was exposed to the exact same brain environment. So that really allowed us to upscale the number of lines that we could interrogate, but also to really reduce the number of mice that we were using. And that was, of course, great both from an ethical perspective, but also from a cost perspective. And because every donor is exposed to the exact same mouse environment, we can use improved statistical modeling, because we can actually really nicely account now for the variability that we observe between mice. Because some mice, of course, may have a bit more amyloid beta pathology or not. Some mice are female, some mice are male, right? So all these kind of variations we can regress out now from the differential expression analysis. So this was basically what the data looked like. So you can see that every stacked bar here is a mouse, and the mice that have this little star, those are the ones that are derived from the APP NLGF mice, so the ones that were exposed to amyloid beta pathology. And so what you can see, and that was also, I think, published multiple times by Michael Wells, is that these pools of cells, in our case microglia, their proportions do not stay equal across these differentiations. So we did pool these cells at equal numbers when we injected them into the mice, but then at 6 months of age, we do see variations in the proportion that each donor occupies within that pool. So this range is roughly from 1% to roughly 30%. But what we were very happy to see is that even though there's these differences between the different donors, every donor is a different color here, we can see that this pool was very consistent across all these different mice. And we also had these 3 anchor lines. So those were the lines named COLF2, BURP1, and L26. And these are always on the top of all the bars. And what we were quite happy to see is that these were always taken along in independent differentiations across these 3 different villages, is that actually their transcriptomic repertoire and also the proportion that they occupied within the entire pool was relatively stable across these 3 different villages. So on average, we had roughly 8,000 cells per donor. And we did have to drop 2 lines because we couldn't find back.
Can I ask you a quick question about this while we're going? So when you see these different numbers of microglia, are there sort of equivalent mouse microglia compensating? Is there a sort of max which is the same in every animal, or is the total number varying?
So there is variation across mice to the extent of what we call graft efficiency. So let's say the number of human microglia that we find back in the brain. But this ranges typically between 60 and 80%. So between 60 and 80% of all microglia that we find in these xenografted brains are of human origin. And so out of those 80%, let's say, of human microglia that we see, within those 80, we see these type of proportion differences. Right. So these proportion differences are very similar across the mice, right? As you can see, there is consistency in the proportions that there are within the same pool. There is some differences in the graft efficiency, but I do think indeed some lines are probably better either at colonizing the brain or they're better at proliferating or survival. This is something that we are not sure about. We have also seen that if we sacrifice, for example, mice at 1 month of age, we see that the proportions are somewhat different. So at that point, they are somewhat more in the same range. So I do think over time it deviates more, right? The ones that are already quite prominent at 1 month of age kind of grow faster, or let's say proportionally increase a bit more than the ones that are already in more limited numbers in the beginning.
One more question then. So this observation that there's a T cell signal that's important in microglial maturation at an early stage, is that would that be a factor in this, that different T cell status in different animals could influence the number of microglia that you find?
Yeah, so what I didn't mention, and that's good that you asked this, is that these mice are immune deficient, so they have no B cells or T cells, and that
great.
need to ensure that they can take these grafts for so long.
Thank you.
Yeah, so that part cannot influence it. So there is also work being done at the moment by Nico Fattorelli, Anna Martinez, and Renzo Mancuso in understanding these microglia progenitors that we obtain, right? So we have this migrate protocol, and at day 18, when the microglia progenitors are budding off from the embryoid bodies, they've done, the idea was actually that, right, this would generate microglia kind of, right, continuously, so that we could harvest these microglia in multiple batches and then graft those in multiple batches for different mice. But actually, we observed that as these lines were, so if we would harvest a week later or 2 weeks later, that actually the proportion of cells that would graft was actually dropping. And so they investigated, right, basically the ontogeny of these cells that we were grafting, and they found that there is indeed a proportion of cells that really mimic these yolk sac derived microglia precursors or macrophage precursors. And so they are likely the ones that really graft and occupy and colonize the brain. And there's also, of course, other types of myeloid cells that are in that dish. So likely, right, the amount of these microglia, well, these YSMP yolk sac derived progenitors that you have will probably dictate the efficiency at colonization. And that will likely drive some of these divergences.
Thanks, that's really helpful. That's great. Thank you.
Okay. Okay, so yeah, in the end, we also dropped 2 lines because we had too little coverage for them. So now that we have this right big pile of data, we have roughly 230,000 individual microglia. We had, of course, some questions that we wanted to ask. One thing was to understand if there were any sex differences between the human microglia. Also to understand whether right, human microglia are in general relatively similar, or whether they are very different already in a wild type brain, in a homeostatic brain when there's no amyloid pathology. And then, of course, we also wanted to understand, right, what is the amyloid pathology response? So if human microglia see amyloid, what do they do? And we know this, of course, already to some extent from postmortem data and also from other xenograft data, but that was always based on 1 or 2 lines. And now we had this, right, 31 lines. So we wanted to see how that, what that looked like. And we also really wanted to understand, are there donor-specific responses to amyloid beta? And so we use this generalized linear mixed effects model, where we assess the impact of donor, of strain, so that's the exposure to amyloid, and also the amyloid times donor interaction effect. And we used mouse as a nested variable, and we corrected for things such as mouse sex, sequencing depth, and sequencing batch. So the first question we asked, right, are there sex differences between the male and the female human microglia? So basically, right, we just separated out all the female microglia from the male microglia and ran the differential expression. And so we saw really stark differences between male and female microglia, with really thousands of genes significantly differentially expressed. And so we could particularly see that the female microglia seem to be much more activated already at baseline. So we saw increased expression of genes implicated in, for example, the MHC class 2 response, but also in viral signaling, right? A lot of these interferon-related genes and also other genes like related to cell adhesion. So there were many genes that were upregulated in the females, also many that were upregulated in males. But in males, I think this was a less consistent result. I think also a lot of non-coding genes that were differentially expressed in male mice. So it was more difficult to annotate their functions. And so because we saw this stark sex differences, but we were also interested to understand, right, what does the rest of the genome do, not only the sex chromosomes, we decided to regress out the impact of the donor sex so that we could look beyond the impact of donor sex on the microglial transcriptome. And so the first question we asked, right, if we take now cells from only the APP human mice, so from the wild type mice, do we see transcriptomic differences between those different lines? And we were actually surprised to see that there were a massive number of differences between those lines. So what I've done here is that basically we plotted the it's a density measure of the space that all these different donors occupy on the UMAP. So where it's darker green, that's where most of the cells for that particular donor reside. And what you can kind of see from this bird's eye view is that different donors occupy different regions in this UMAP. So already showing that from a transcriptional perspective, from a bird's eye transcriptional perspective, these cells are very different. What we did for the differential expression analysis is that we calculated a kind of average microglia, so the average response across all these different donors, and then calculated how is each donor different from that average. So when we did that, we really saw that there were hundreds to thousands of differentially expressed genes. So meaning that, even in a wild type situation, microglia coming from different donors are really vastly different from each other. And so given that these numbers were so large, we were a bit worried that this may be due to our statistical model. So we also ran some extra analyses to be sure that these were not false positives. So Dan Mughars, who's the bioinformatician that did all these analyses, he basically made like a random model where he kept all the covariates similar. So the amount of cells that were derived from a donor, for example, or the mouse it was derived from, those were all kept stable. But then he randomly assigned a cell to a donor. So to kind of see, if you then do the differential expression analysis, you should really see no significantly differentially expressed genes. And that was fortunately also the case. As you can see in the middle graph, we really get 0 to 1 or maximally 2 differentially expressed genes when we take this kind of randomized data set. And also for fun, he also tried the same using a Wilcoxon-based differential expression. And so you really see that with the Wilcoxon-based differential expression, you get this massive amount of false positives. So we were quite sure that the number of genes that we found to be differentially expressed between those donors was correct, and they really were in the hundreds to the thousands. Because we had so many differentially expressed genes, we wanted to understand a bit better what's going on between these donors, how are they different from each other? And so very often people use these clustering-based algorithms, so where they assign microglia to belong to certain cell states. But this really draws quite harsh lines, between microglia, and really says, they're either homeostatic or they are disease-associated microglia. But microglia are, of course, very dynamic, and depending on when you harvest the microglia or when you observe them, they may be more or less homeostatic, or they may be transitioning between these different states. So we wanted to use a different way actually to gain insights into the data. And so for that, we use this WGCNA algorithm, which I'm pretty sure you're familiar with. So basically, this clusters together genes into these gene modules. And those gene modules, by and large, represent biological processes that are ongoing. And so by using WGCNA, we had these 12 modules that we found that can describe how microglia are behaving. So, for example, we find many clusters that are really kind of form a continuum of gene expression across the UMAP. So, for example, this green module, which kind of represents homeostatic microglia. But you can also see that some modules are really restricted to some regions of the UMAP, like this blue module, which is the interferon response. And I also want to point your attention to the brown module and the magenta module, as they are this kind of disease-associated module that was described earlier, and so contains a lot of lysosomal genes and endocytosis genes. And also this magenta module, which is the MHC class 2 response. And that will become quite important also during the rest of the talk. So now that we had all these different modules, we could basically score how every individual donor was, right, whether they had genes that were upregulated within that module or downregulated within that module. And so what you can see in this graph, which is quite busy, is that you can see all the different modules on the bottom with their different color codes, and every donor is on the y-axis. And so basically, if you eyeball right over the different rows, you can see that the pattern for which modules are enriched and are depleted, right, or where genes are upregulated or downregulated, you can see that that's quite different across all these different donors. So basically showing that every donor really has a unique profile of genes that it's up or downregulated when it's present in a homeostatic brain, right? So even under physiological conditions, there's already a vast diversity across all these microglia coming from different donors. And so I particularly want to focus your attention on these magenta modules, so the MHC class 2 response, and this brown module, that's this kind of DAM, so disease-associated microglia response. And what was very interesting to see is that if we plot this a bit differently, you can see that there's really vast differences between the expression levels that certain donors have compared to other donors. So for example, this L23 and L25 donor really show roughly 3 to 4 times more MHC class 2 expression, and other donors show very high DAM expression, whereas other donors show hardly any induction of these profiles. But what is interesting is that these profiles, so MHC class 2 expression and DAM expression, those have often been linked to pathology. So these programs are often only initiated when a microglia sees pathology. Yet we see these programs already highly activated in certain donors under complete physiological conditions. So when there's no amyloid beta or other stimuli basically to respond to. And so this is interesting to us because it really also questions, right, what is a homeostatic microglia? That concept, which was always attributed to expression of certain homeostatic genes like P2RY12 or TMEM119, right? These kind of homeostatic profiles may be very different for individual donors. And so kind of, right, what is your baseline or what's your set point, your initial set point may be very different depending on your genetic background.
Can I ask you another question here? A spectacular result that you're finding, but is there such a thing as a canonical microglial signature hidden in here? Is this variation happening in a subset of modules, and other modules are stable? What is a microglial cell? Is what I'm wondering about.
Yeah, so I think that's a very good question, right? Because I think very often and this is often based on data coming from 1 or 2 lines because, for example, right, Renzo Mancuso in collaboration with Bart, they've done already some xenograft models as well as Matthew Burton-Jones at UC Irvine. Right, and they indeed do describe that there are right, that most microglia actually, when there's no pathology to respond to, they reside in this kind of homeostatic state where, right, they express certain marker genes. Yet, I mean, we do find a homeostatic module that's actually the green-yellow module here named motility. But you can already see that there's quite a lot of variation already in this homeostatic module. So some microglia express it much less than the average, and other microglia express it and other donors express it much more than the average. So I think, right, even under homeostatic conditions, yeah, what is kind of the baseline microglia profile is very different depending on your genetic background. Does that answer your question?
Well, it's very, very cool result. I mean,
yeah.
And are the modules mutually exclusive? I'm
No.
wondering how much we yet have to discover here.
Yeah, no. So in principle, a microglia can score high or low on multiple modules, so they're not exclusive. We do see that some are a bit exclusive. So, for example, this green yellow module, so that kind of homeostatic module, anti-correlates quite starkly with the expression of the magenta and the brown module. So they seem to be quite opposite sides of the spectrum, right? So either you express homeostatic genes or these DAM or magenta genes. So we do see some specific correlation patterns between certain modules, where certain ones correlate more strongly and other ones anti-correlate more strongly.
Maybe I could just ask one other very general question just to keep everybody on board.
Yeah.
So if when you think about other data sets that people have analyzed using these same kind of tools, and they classify cells as different kinds of neuron, do you think that they're missing a whole lot of variation, or that microglia are really unusual in showing this kind of heterogeneity between donors?
No, I don't think microglia will be unusual. I think this will also be the case for other cell types, right? I think this is probably right. I mean, we also assume if we even look at people, right, we know people look differently. So it's in that sense, it's naive to assume that that this kind of diversity is also not present on any body cell to an extent. I think what we were indeed a bit surprised about is the extent to which the differences, right? The magnitude of differences that there exists between these cells, and particularly that certain programs that we associate with disease can already really be expressed under these physiological conditions. That is something I think that we didn't expect.
It's extremely cool. Thanks.
Yeah, you're welcome. So the next question we asked, right? So in general, how do human microglia respond to amyloid beta pathology? Now looking at this from a larger cohort than just one or two lines, but really looking at these 29 donors that we had in total. So basically, we extracted all the cells coming from the human mice and compared them to all the cells coming from the APP NLGF mice to really see which gene expression profiles are up or down regulated. And so this basically reinforces a lot of the literature that's already out there, where we can see that if microglia are exposed to amyloid beta pathology, we see this upregulation of this magenta module, so the MHC class II module, as well as this brown human DAM module, which has a lot of lysosomal genes and a lot of lipid genes. So in essence, this data was not surprising, but it was nice to see that it replicates. So in general, if human microglia are exposed to amyloid beta pathology, by and large, they will induce this kind of response. So upregulation of these lysosomal genes and these MHC class II genes. We also confirmed this upregulation of the MHC class II response by flow cytometry, so on the protein level. And we could also actually find that many of the co-stimulatory and co-inhibitory adapters that are often found on antigen presenting cells, like PD-L1, CD80, and CD86 were also upregulated in human microglia when they see amyloid beta pathology. So we kind of explored this a bit further, this MHC class II response, and we saw, I think this was also for sure described before, that this MHC class II and DAM response really occurred mostly around amyloid beta plaques. And we could also showcase that in postmortem brain from AD cases. So we see this massive upregulation of MHC class II in microglia surrounding amyloid plaques. And we did not see that response so much in a non-demented control brain. So, of course, if we say MHC class II, very often, we think about antigen-presenting cells and interaction of antigen-presenting cells with T cells. So this is kind of a cry for help for T cells to come and come and help in clearing this pathogen. So we were interested to understand, because of course we have mice that are devoid of T cells, so we cannot really look at this T cell microglia interaction within this mouse model. So therefore we reached out to the lab of Max Mazzone and we performed a T cell co-culture assay where we extracted human microglia coming from either APP human mice or APP NLGF mice. And we co-cultured those with naive T cells, both CD4 T cells as well as CD8 T cells. And then we saw that indeed, when human microglia are exposed to amyloid beta pathology, they express this MHC class II profile. And then we could also see that they are then able to activate and induce proliferation in T cells, and also to stimulate cytokine release from T cells. And that was both the case in CD4 T cells as well as in CD8 T cells. But what is interesting is that, right, in our system, of course, we cannot study those T cells, amyloid beta interaction, microglia interactions. But what is also interesting is that actually in the Alzheimer brain, there's still some controversy on this topic, but there's not a massive influx of T cells into the amyloid brain. So we can see, right, that even though some T cells may reside in the brain, they are very often in this perivascular space, and then not very often found really deep into the brain parenchyma. But it's not like in MS where there's this massive T cell influx to come to the aid of microglia, despite the fact that these microglia are signaling with MHC class 2, right, for these T cells to come. So that is something we're still kind of chewing on. So of course, next question was, right, we know now that these microglia can upregulate MHC class 2 when they, and DAM profiles when they see amyloid beta pathology. But so how do now different donors actually respond to amyloid beta pathology? Do we also see the stark differences as we saw in the wild type in the physiological context? And yes, we do. Again, we kind of plot here all the cells coming from the APP and LGF mice only. And we can see here this UMAP representation of each donor. So again, you can see, right, the different donors really occupy different spaces on the UMAP. And again, when we use our WGCNA clustering algorithm, right, we can see again that individual donors show quite different profiles in response to amyloid beta pathology. And again, the MHC class 2 module, the magenta module, as well as the brown module showed the starkest degrees of variation across these different donors. And so what you can see in this graph is that actually this little red dot represents the average amyloid response. So that is the amount of MHC class 2 expression that's induced on average by human microglia when they see amyloid. And then you can see that certain donors really deviate approximately threefold above the, so they deviate like by threefold from the average response. So they induce roughly 3 times more MHC class 2 genes than the average response. And some actually fail to induce these responses at all, right? So this UP6 line, for example, hardly induces any MHC class 2 and also hardly induces any DAM profile when it sees amyloid. So again, highlighting indeed that also under this kind of amyloid stress, depending on your genetic background, right, your microglia may respond very differently to amyloid beta pathology.
Can I ask a quick question on that previous figure? So do you find any commonalities between the lines that are behaving similarly? So for example, like BR21 and BR33 seem to exist in the same space in the UMAP. Genetically, is there anything that you can pull out between those lines that behave similarly in response to the A beta challenge? For example, you know, can you stratify them by their TREM2 R47H status or their ApoE4 status?
Yeah, very good question. Indeed, that was something, of course, that was also something that we really wanted to understand, right? Can we delve a bit deeper into this genetics? That was a bit of a daunting task. So what we did is actually, I'll get to that in the next slide, is that we utilized again these polygenic risk scores that we calculated all the way at the beginning, and we used those polygenic risk scores at the time to select our line, so that we had this kind of a spectrum of polygenic risk score. And so the first thing actually that we did was to see if we now have this magenta module expression. So that's right, that's this graph. Do we see differences between cases that we know are AD cases and individuals that we know are non-demented control? And we could actually see that Alzheimer cases showed on average a higher expression of this MHC class 2 response than non-demented controls. But what was really interesting is that we could actually correlate the level of MHC class 2 response when seeing amyloid to their polygenic risk score that we calculated previously. So showing that at least part of the risk for Alzheimer's disease probably funnels into how microglia respond to amyloid beta pathology. And maybe I'll just quickly pull up these slides as well, because now that you're asking. So We also checked this response, right, because we have this magenta module, of course, in response to amyloid, but also in the wild type situation. So actually, when we tried to correlate this polygenic risk to the physiological MHC class 2 response, there was no correlation. And also we tried to see if we only pull out the SNPs that are in this, that span the genes that are in this magenta module. So those are all the MHC class 2 genes. Then we actually also don't see any correlation. And we also cannot link the MHC class 2 response to amyloid to any of the known MHC class 2 haplotypes. So, right, that kind of makes us want to believe that indeed this kind of response, this MHC class 2 response, is very polygenically driven. So it's not driven by SNPs within the MHC class 2 locus, but it's probably driven by, right, SNPs basically everywhere else in the genome. But we haven't been able to kind of pinpoint yet on particular genes that are really driving this expression. So that's something that we're still trying to battle with a bit. I hope that kind of answers your question.
Can I ask a related question?
Yes.
I know it's kind of hard to do this because it's like all in the all mashed together sort of, but do you think there's any contributions to like where those microglia ended up within the tissue, like how close they were to an amyloid plaque versus like, some might be more migratory, so maybe they're migrating faster to the plaques, or how where they're localized within the tissue may impact some of these gene response changes that you're seeing?
Yes, so indeed, that's something that we don't know at this moment because indeed we've extracted all the microglia from the tissue, so we don't have their spatial locations. I think based on the transcriptome and the fact that in general, when microglia see amyloid beta, we see this upregulation of DAM and MHC class 2 genes. That is a profile that we can see back in the different lines. And also something that I haven't shown now is, but we also have this, so there were also 16 lines we actually grafted individually. And so for those lines, right, so they are grafted by themselves in a single mouse, and they were grafted in the amyloid mouse. And actually, if we look at their transcriptomic profiles coming either from, right, being individually grafted or coming from these villages, their transcriptomic profiles were very similar. So, right, so basically, if a microglia is exposed to amyloid beta pathology, it will induce a particular program. We also see those programs in the villages. So in that respect, I can probably conclude that, right, all microglia that we take from the mouse brain have been exposed to amyloid at some proportion or another, right? Maybe some will be more present around plaques. Indeed, there may be a difference in the way they proliferate or they migrate. So yeah, I can't control in full, of course, where within that village, right, all these microglia end up. But the fact that the data from the single grafts is roughly similar to the ones from the village grafts do make me quite confident that at least all microglia have been exposed to plaques, and that yeah, these responses are genuine and not driven by the village, let's say.
Sorry, maybe I missed you said it, but do you see any relationship with the lines that basically took over the mix, the one that were the most proliferatives, compared to how their microglia behaves?
In how they behave, you mean, in terms of
Yeah,
their
compared to this
profile?
phenotype that they're
Yeah. No, so our statistical model does account for these differences in distributions or in proportion. Basically, what it does is that, right, if we have more cells of a particular line, it just gives us more power to detect differences. So I think but the differences that we do pick up, I believe, are really there. So and I.
But could it be that basically the fact that they took over is physiological and they behave a certain way then in your WGCNA, not technical, right? Something that could be biological relevant. That do they behave the same way, the three more proliferative in your WGCNA?
Yeah, I think so. I need to go back to this, but I think we didn't see any. So, for example, what we also did is that, for example, just a basic PCA clustering of all these lines, just to see if we could find some kind of similarities right across these lines. But they were really like a blob, right? So they were basically spreading everywhere throughout the PCA. There were no lines that were clustering more together. If we could have seen these kind of clusterings where lines would have been more similar, right? Then indeed these kind of things like right, their proliferative capacity, or maybe whether they were AD case or control, or any other phenotypes that we knew from these lines, we could have picked up, but that we didn't really see, right? They were all kind of similarly different from each other.
I see. Thank you.
Yes. Okay, that kind of brings me to the end. So I hope I've convinced you, right, that these human microglia villages are a really nice way to understand the impact of complex genetics on microglia, but in an in vivo setting. I've also shown you that female microglia appear to be much more activated, upregulating these interferon and MHC class II responses. I've also shown that genetic heterogeneity really profoundly impacts the human microglia transcriptome. This was most evident for this MHC class II module and this human DAM module. And we saw this both under physiological as well as pathological conditions. And that we were quite surprised to see how different those microglia already behaved under homeostatic conditions. So that indeed we are wondering a bit, right, of what is actually a homeostatic microglia, and that's probably very different for each person. And I hope I've also convinced you that right, at least some of the genetic risk for Alzheimer's disease may shape how human microglia respond to amyloid beta pathology. And something that we're still pursuing is now to understand a bit more what this MHC class II response actually means for microglia. Is it really driving a particular profile or the different responses of microglia towards amyloid beta plaques, or is it more of a tag that it's in a particular state or doing something right, or maybe a tag for a particular dysfunction of the microglia? So that's something we're actively pursuing at the moment. And happy to take any further questions. Thank you.
Thanks so much. A lot
You're
to think
welcome.
about.
Yes, yeah, sorry, it may have been a bit fast because I do tend to ramble.
Spectacular. I don't think you're going to get away with being self-deprecating after a talk like that. I mean, there's lots of things to ask, but maybe just to get the ball rolling, to come back to this like central finding, which I think is really impressive. Different people have very different microglia, at least under these conditions.
Yes.
And so it seems to me there's sort of like two versions of that question. One I already sort of asked, but I'd like to ask it again, which is, do you think that when people normally do their informatic analysis of transcriptomes, they're missing a lot of this because of the way they're using clustering tools and a kind of pre-existing idea of cell type to classify cell states? And that there really is much more variability than we're recognizing? That's one question. I sort of asked it before, and you said they're missing it, you seem to say.
Yes, so I think, right, I think we are aware of heterogeneity when we do human studies, right? In postmortem studies, I think we often acknowledge that, but of course there's I think then also this interplay of right, kind of the disease process of a person, right, the agony of dying, what that does to the transcriptome, right, the individual differences based on genetics, how the human microglial transcriptome also changes depending on the pathology it saw. The differences between people in terms of, right, the amount of pathology that they have, right? So I think if you look at postmortem tissue, there are so many of these different kind of factors, right, that play a role there, and right there, and also technical issues that I think it's very difficult to kind of tease that apart, right? And that is kind of of course, the nice setup with this is that we can really tease those different aspects apart, right? We can transplant them or expose these microglia to whatever we want, depending on what type of mouse we can put them into or what we can subject the mouse to. So I think it's this kind of village approach that was really inspired by the Eggen lab and the Macarel lab, that really allowed us also to address those questions. And I think it will be difficult to kind of to really tease that apart from a postmortem perspective.
Sorry, can I ask a question that builds on
go
that?
for it.
Jump in.
Yeah.
So I think we're kind of trying to get to understanding cell state versus individual variability. And so I wonder, like at baseline, how repeatable, when you put in the same from a different batch, the same cell lines, like how repeatable are those differentially expressed genes in your microglia just at baseline? I know you said that in the disease context that your 16 cell lines that you sort of individually transplanted, those seem to be quite similar to the disease state when they're in the village. I just wonder, like baseline, what is the kind of repeatability of these differentially expressed genes?
Yeah, so the only answer I can give to that is we did. So we had these 3 anchor lines that we took along in all 3 villages. So those villages were created independently. So those were 3 different differentiation batches. And those 3 anchor lines that we took along, we did compare them also, how do they behave across the 3 villages, both in the wild type situation as well as in the amyloid situation. And we do see roughly, I think 70 to 80% correlation. So in the differential expression, even at baseline. So I do think in that sense, the transcriptome, of course, there's variation because there's differences in differentiation batches, and I think everybody working with iPSCs acknowledges that. I do think what really helps with the xenotransplantation is that you kind of force microglia into a certain environment, So that really helps to shape them and kind of stabilize their transcriptome. So I do think in that sense it's relatively reproducible, The transcriptomic repertoires that we see and this variability at baseline that we see, I do think is reproducible. It's not it was not a one-off experiment. Yeah.
Thank you.
You're welcome.
Super interesting listening to you. Can I ask you just to comment on another set of data that I don't know as well as I should, but haven't people done a limited number of biopsies in Alzheimer's brains? And do they see a similar kind of variability that you see here?
That's a good question. That's this Finnish cohort that you're referring to, I think, with this normal pressure hydrocephaly cases.
Yeah, possibly. I'm not up to it, but I should look it up.
Yeah. So we haven't analyzed that data from that perspective, but that would be an interesting way to look at things. But again, I think there is this problem with kind of working from human data in I think this where we're still, even though you have their genetic signature, I think that depending on the environment that they're in, these microglia also show different responses, So I think there may be ways actually to normalize that out, just to look at their amyloid beta load and kind of subtract that from their transcriptomic, from the rest of their transcriptomic response, and then to see what additional transcriptomic differences there are. I guess we will be less certain to understand what is driven by genetics and what is still driven by other factors. But it may be a way in to see how different this is. To comment also on the clustering, so I do think that in general, people have struggled to find, for example, this human DAM module or a profile in human microglia. So they find it to some extent. It's not exactly the same. Also, different data sets kind of show different profiles. So there has been quite a struggle to kind of integrate different postmortem data together and to really find these kind of definitive clusters. And I think indeed the genetic heterogeneity that consists does not help, let's say, to find these kind of consistent behaviors. Well, we can maybe question if they are even there.
Yeah, it's quite clear in the mice, less so in the humans. Yes, Sally and Alex, we have 2 hands up. Jump on in, guys. Hi.
I think Alex is probably before me.
No, but please go ahead.
Okay. It's a quick question. It was such an amazing talk, and I would thank you so much. I'm wondering, what you think about now, or how we think about studies. Most studies don't use 31 lines. They use, 3 or 5 lines. What do you think about the picture that we're getting when we are looking at a smaller subset of lines? How how do we kind of put our arms around this?
Well, I mean, right, I think to be honest, right, we also don't do all our experiments in the lab with 31 lines, right? So this yeah, I think that's a struggle that, right, it's just often too massive amount of work. So yeah, I do think, right, we need to I think it is important, right, that we try to replicate these kind of findings that we do across different individuals, right? And of course now I've looked at these 31 lines, which are still, for example, only Caucasians, right? It would also be interesting to see how other genetic backgrounds, right, with people from with a higher genetic diversity would also respond. Right. I think in that sense, there's kind of endless variability that we need to take into account and look at.
Yeah, I just feel that it makes me think that unless you're really looking across a diverse cohort, you could be making conclusions about how microglia behave based on a limited set of donors, and that really may not reflect,
No.
accurately the range that you see.
No, I agree. And I do think that this kind of right also really provides a basis for when we think about pharmacodynamics as well, right? When if we start to treat,
So
I
true.
think it would be great if right that could be kind of assessed on multiple different individuals, because indeed depending on your kind of your baseline, right, or your set point, you may respond very differently to a treatment. So yeah, for sure, this heterogeneity is important, and we need to research it more. But yeah, I'm also aware of the costs and the effort, right, that comes along with it. So yeah,
Thank you.
you're welcome.
Yeah,
Yes,
hi. That was related also to the previous question that were asked. I was wondering if you also collected for the same IPS lines village at the IPS states. Is this been done by you or others for the same IPS village, and see if notably the marker you see between individuals are like either predicted by IPS states or like already present in some form, or is it only specific to microglia what you saw? And to compare if the state in IPS can predict how they behave in the after implantation, and also if you collected before implantation, also the single cell.
So all the answers are no, unfortunately no. So we haven't looked at the iPSCs. We haven't related iPSC transcriptome to the microglial transcriptome. Also, we haven't done single cell on the, let's say, the injectate. So before, right, on the human microglia precursors. Indeed, these are all things that could still be done and could indeed be informative. We have looked into, but that was more from a technical point of view, to, for example, already merge the cells at the iPSC stage before we start making the embryonic bodies. That was not so successful because I think that was also shown in some of the research from Michael Wells and also others, right, that indeed iPSCs have different rates of proliferation, so that, right, in the end, often one takes over also in the culture. Yeah, so no, I haven't done all that research, but that's still also really of interest.
And so do you think those inter-individual modules that you're seeing can also be applied to any, I think you said, to any cell types? Like
yes, for sure.
Find the same in IPS or on neurons?
Yeah, so WGCNA is a common tool, let's say, right? And it's just driven by the gene expression data that you put in. I mean, we have used WGCNA also on different sets of microglia in the lab, and we do roughly see the same modules come out. But of course, if you work on neuron data, those modules will look very different.
Thank you.
So I have two comments that might broaden our discussion a touch here. One would be you're examining directly variation from the point of view of microglial reactivity here. But on the environment end, you have a wild type mouse that is humanized APP, and then the other APP model to have a pathology versus non-pathology environment. Just as a very general point, we could even think about diversity on that end as richly as we are on the reactivity end, because just like everyone's microglial reactivity will be different, the generation of pathology presumably is not the same, unless it is simply the presence of plaques that is the key to everything and it just sets it off. The second question would be, and this is thinking broadly in thinking about Alzheimer's and therapeutic strategies for it, where there are the vast majority of advanced therapeutic candidates for AD are pathology based. Can you clean up A beta, right? Antibodies to it and so on. What does all of this mean? Meaning your work as well as more broader perspectives. What does all of it mean if pathology alone isn't the key and resilience of some kind is? And I say this thinking about a lot of recent observations where if you look at a control in an AD cohort in a postmortem brain collection, there are a huge number of controls that are full of amyloid plaques that don't have cognitive impairment. And so to complicate, if this weren't already complicated enough, how does that complicate our interpretation of what everything means in both pathology and response to pathology if it is not necessary and sufficient for disease to have a plaque in your brain? And this is kind of for everybody, but
Yeah, I'm trying to think how to answer. So just out of maybe a curiosity side note, so we are also collaborating now with a woman called Henne Holstegen. So she has a lab in Amsterdam and just also joined the VIB in Leuven. So she works on people that are 100 years.
Yes, this cohort I've heard of. It's crazy, right? They're super old.
Yeah,
Yes,
the
tell
100
us
plus
about it.
cohort. So they are all people over 100 years of age that are cognitively still intact. So at inclusion, right, they still have to claim themselves that they are cognitively intact. They are verified by family members, and then if they are included, they are followed up right until they die. Roughly a third of them also donates their brain. And so they have all this right very rich neuropathology data as well as as well as all this cognitive data. And so actually she also made iPSCs from some of these centenarians. So we're now actually also in the process of doing research on those as well, also using these village approaches.
Oh, wonderful.
Yeah, because actually so in her mind, she says that actually amyloid beta is not a benign consequence of aging. So she sees that if there's amyloid, so most of these centenarians, it's really interesting, they have a much lower plaque load than actually, than you would expect. And also, right, so most are still cognitively intact. They have a lower plaque load. So they're probably, but they do see that sorry, I will say this better. So what they see is if you look at their TAL phases for plaque load, which basically looks at the spread of amyloid beta pathology, then they are at the same level as an Alzheimer case. But what they see is that it's not per se the spread of the amyloid beta that's different, but it's particularly the load. So they see that even though the amyloid may have spread to different brain regions, there's far less amyloid beta pathology than you see in AD cases, and especially given their age. Also, they see that most people kind of avoid the transition to tau pathology. So even though there's amyloid pathology, right, there's little transition to tau pathology. And if they do transition to tau pathology, then of course there's often also cognitive decline. But she does also see that those that have a high amyloid beta load, that they also cognitively decline. So she thinks that, right, in general, having amyloid beta is not a good thing. It still is detrimental to your brain. There's probably some threshold, right, that you can keep, and then you kind of, right, you start inducing tau pathology and you fall over the cliff. But indeed, right, some of their resilience probably also goes through the fact that they can contain this amyloid beta pathology. And that may be right either due on the level of the neurons, where the neurons maybe produce less amyloid pathology, or at the microglia, right? Because maybe the microglia are better at containing the plaque load. But of course, some resilience may also be in the neurons because the neurons are still exposed to some level of amyloid stress, but they don't transition to tau pathology. So, right, that I think that kind of genetic signature of these individuals, because they are also much depleted for Alzheimer risk loci, right? That kind of whole genetic signature of these individuals gives them resilience, and through which pathways? That's still the $1 million question, right? Does it go through better neuronal function, through better microglial function, less pathology buildup, right? And how is that instigated? I think that's still a question. And then your first part, I forgot. Your
The
first
reactivity, so variation on the environment end, but that's
yeah,
kind of a wide open one.
Well, but you could ask a very specific version of it, Carlo. Are you planning to use the same sort of approach on another mouse model, say a tau pathology mouse model?
So initially, yes, but then at a certain point, we decided to kind of limit it to the amyloid load. At the moment, we're not planning to do it because, well, I know there's a lot of literature involving right how microglia may relate to tau pathology. But in the Stoper lab, we were quite, how do you say this, kind of fixed on the idea that microglia may mainly impact amyloid pathology and not per se tau pathology, let's say directly. So that's why we kind of stuck to the amyloid pathology at the moment. But it is still something that, right, it's kind of on the to-do list. But I think there's many things still on the to-do list. So I'm not sure we'll get there.
Thank you.
I can ask another question, maybe
Yes.
just more. So this is more sort of futuristic, in terms of what you would do next. So if you were to sort of target specific cell lines because of their expression profiles from what you've revealed, for later functional assays, like what would you think about, targeting next and sort of, characterizing next in your model?
Yeah, so something that we really want to understand now in the lab is this MHC class 2 response because we're quite puzzled by this because right it's so these microglia kind of seem to uptake this role of antigen presenting cells, right? They upregulate this MHC class 2. They also have all these co-stimulatory and inhibitory receptors. We have some data in the lab that shows that, right, they are really actually presenting antigens. Yet there is in the AD brain, at least right to my knowledge, if I look at the literature, right, there's no vast influx of T cells into the Alzheimer brain. So I think, right, and I'm not saying that therefore T cells are to be disregarded altogether. I'm pretty sure, right, they play an important role, but I'm not super convinced that right, the MHC class 2 response is really solely there to cry out for T cells. And so I really wonder, right, what that signals. Why is that important, that MHC class 2 response? What, right, how is it signaling to the rest of the brain? What is it doing to other cell types? So this is something that we want to tackle a bit more and understand a bit better.
Yeah, I think I've seen some recent work where it's in dealt with some papers recently that, we're seeing these MHC complexes upregulated in early development, like in neurons expressed in places that we didn't really. So I think it's an interesting part of cell identity that we don't really fully understand, as opposed to, necessarily immune function, strictly speaking.
Yeah. No, I agree. Indeed, I think so. There's also some literature on astrocytes expressing MHC class 2. Indeed, and some of these more immune modulatory receptors are also expressed on neurons. I'm not sure about MHC. Well, indeed, the developmental part on neurons I wasn't aware of. So yeah, I think indeed it's something that I really want to explore a bit further and understand further. So.
Great.
Well, thanks a lot for all the interesting questions. So it's really nice exchanging some ideas.
Thank you. And thank you for being up in your evening away from your
Sure.
kids.
No problem.
So something I'd like to mention to the group, actually, I was talking to Annerieke about doing a little bit of a data session on issues that are very close to this. We have a Nemo Analytics online public web portal that in which we've pulled in a lot of Alzheimer's, both bulk RNA sequencing and single cell RNA sequencing in both mouse models and in human tissues. And we were going to go look through some of that. And so just to give you a little bit of an idea of what's there, we can take signatures of DAMs from their original publications. And Annerieke, I'd love to get your gene lists for the modules you just shared so that we could look at these across mouse models and human tissues. And, we could end up making this kind of a DDD-centered session too, an Alzheimer's DDD session. But I'll send out some stuff as we talk more. But if you're interested in that, look out for emails coming up, and we can have kind of a data-driven discussion to see what's out there in both the data world and then the signature world to see, like we've been talking about all along, what mouse models show it? Is it across all the mouse models? What are these things are showing up in the human data? Is it across all humans? Is it just some of them showing these things? So we've just now started to bring in the data explosion in Alzheimer's is huge, but we have a lot of the most recent big studies in that we can start to look at these signatures in. So that might be a lot of fun to do together.
Yeah, it's really interesting. So also, I know Phil de Jager also has this kind of work group around, well, that's more on like specifically on microglia signatures. So they're also trying to find some kind of consensus signature, right, across all these different data sets, to see what the consensus is in terms of microglial expression. I'm not fully following, but I know that, right, that's an ongoing effort.
Yes, that sounds like something we'd be very interested in looking at. Wonderful. I'll try to dig into that some too. Thanks so much, and I'll be in touch about the data session.
Great. Thank you so much.
Hartelijk
All right.
bedankt.
Ja, graag gedaan. Bye.
Thank you. Thanks very much.
Very welcome. Bye-bye.